I'll do a quick intro of Hans, who I'm sure everybody here knows. I guess you probably have had almost everyone in the class, this class in one of your classes, Hans. But Hans will tell us today about give us a primer really in metalloenzymes and some of the exciting developments there. Just quick background. Hans, this is one that I can do from memory. Although if I screw it up, now having said that, I'm going to be embarrassed. But Hans did his undergrad work at Columbia with Tristan Lambert. I think was part of the very early Lambert crew, maybe one of the first undergrads to join the group, if I'm not mistaken, and then moved to Scripps to join the Barron lab, where he completed a landmark total synthesis of wobogenin using a redox relay strategy, moved to Francis Arnold's group and worked on a lot of interesting carbene and nitrene transfer reactions using directed evolution, and then has launched an independent career on the Florida campus starting in 2016, and has really crafted a totally unique program. I think charting out a path forward in total synthesis and thinking about how to use strategic biocatalytic disconnections, biocatalytic reactions and to enable new disconnections, and synthetic planning obviously has made a big impact on the field, won a number of awards, including NSF Career, Sloan, and recently the Natural Products Report Emerging Investigator Award, and was deservingly promoted to associate professor very recently. So today Hans will be putting on his biocatalysis, wearing his biocatalysis hat and bioinorganic hat. And this is a big field, so it's hard, I think some departments will have bioinorganic essentially as a whole class. And so he'll try to cover a lot of ground, but hopefully everyone recognizes and appreciates that it's just such a big body of work that there are some things we'll have to go through quickly in the interest of time. So I won't take any more of your time, Hans. Feel free to take it away.
All right. Thank you, Kiri. Okay, let me share my screen. And switch to laser pointer. Okay. All right. As Kiri mentioned, I'll be covering or providing an overview of the different types of natural and artificial metal enzymes and the types of catalysis that you can perform with them. And as the name suggests, metal enzymes refer to enzymes that contain metal cofactors. Of course, this is a very broad term. The metal cofactors itself can either be bound to the protein directly by ligation of some of the side chains of the protein residues, or they can be bound through the use of prosthetic groups. So prosthetic groups would be like this porphyrin here in the P450s. And this slide gives an overview of some of the types of cofactors that exist in natural metal enzymes, and you can see that the cofactors is very diverse. So we'll start with oxygenases, and I guess before going to some of the details, I want to cover an important definition in the oxygenase superfamily. So you have monooxygenase and dioxygenase, and the difference is that for monooxygenase, only one oxygen atom from dioxygen will be incorporated in the product, and the other oxygen atom from dioxygen will be reduced to water. For dioxygenase, both oxygen atoms will be incorporated in the products from the substrates. So so you won't get water from that. And on the left-hand side here, you see several different types of oxygenases. You're probably the most familiar with the P450s, which are this iron porphyrin containing enzymes. And then there are also iron-based enzymes that don't have porphyrin rings, and this can be found in the risky oxygenases and non-heme deoxygenases. And we'll cover these three superfamilies later on in the lecture. You also have this methane monooxygenases that can hydroxylate methane. So it's a pretty remarkable reaction. The one issue with this enzyme is that these are membrane proteins and these are typically very hard to work with. So I think the applications have been quite limited. Oxygenation reactions are not only limited to iron, there are also copper-dependent enzymes that can catalyze oxidation. I think these are typically found in higher species like plant and fungi, and this is one example of copper-dependent monooxygenase that contain this 2-histidine, and I guess in this case it's a dicopper enzyme that has multiple histidine binding to the copper. And in addition to oxygenases, there are also other metal enzymes that serve other functions in nature. Iron sulfur clusters are very common types of proteins, and these are typically involved in electron transfer in various oxidation and reductant processes. One thing to note about iron sulfur clusters is that they are almost always air sensitive, so they can be pretty hard to work with. We also have this an enzyme that belongs to the ribonucleotide reductase superfamily, and this superfamily is involved in the conversion of ribonucleotide to nucleotide. And as you may know, ribonucleotide would eventually become RNA, and then deoxyribonucleotide will become DNA. So this enzyme is involved in the regulation of the rate of DNA synthesis in nature. So it plays a pretty important role. We're going to move on to more exotic cofactors that has rather very interesting ligand structures. You have this hydrogenases that's involved in a reversible conversion of hydrogen to proton and electrons. And then you also have unusual metals like molybdenum or nickel. So you can find molybdenum in xanthine oxidase. This is involved in the conversion of oxidation of hypoxanthine to xanthine. And this nickel enzyme is discovered quite recently, I believe. And so it has a pretty unique pincer structure, and it actually is involved in the interconversion of the different stereoisomers of lactate. And I guess the granddaddy of them all is this Firmoco nitrogenase that's involved in the conversion of nitrogen to ammonia. So it's important in nitrogen fixation in a lot of living species. And you can see that it has a very complex cofactor structure. It even has a carbon atom in the middle of the cofactor. And the biogenesis of this carbon atom is quite fascinating if you want to take a look at it. And as Kiri said, this is a very broad topic, and there's an entire field of inorganic chemistry where people would study all the details of the reaction mechanism of some of these enzymes. And I guess just to give a brief overview of the type of techniques that you can use to study these enzymes, there's a whole range of spectroscopic techniques that you can use, and each of these techniques operates in different energy range, and their use will depend on the types of transition that you want to study within the species. And I guess the important thing to note is you want to match the energy range of your source with the transition energy that you want to study. So it can range from gamma ray and X-ray that you can use to, I guess, to study the transition in the core electrons, such as transition from the 1s electron or 2s electron to the continuum. And I should also mention that for most power, this is only specific to ion species, and you actually have to use ion 57 ion 57 isotope to perform this technique. You can study electronic transitions by using UV phase spectroscopy. And of course, you are probably aware of the use of infrared and Raman spectroscopy to measure different types of vibrational modes. And Going to things that are lower in energy, you have EPR and ENDOR, which are used to study electronic transitions between different spin states in your species. So these are, I guess this is just a very superficial overview of the types of spectroscopies that are available. And I guess if you want to, you can ask Kiri to bring a more qualified expert to teach you about the fine details of these techniques. Okay, let's move on to some of the enzymes that I'll be covering. Specifically, those I'll be covering those that have found applications in biocatalysis. So I will start with the P450s, which are some of the most extensively studied metalloproteins in the literature. I mentioned before that these are heme-containing enzymes, and so it's defined by the presence of a heme cofactor, which is also known as protoporphyrin 9 here. And it also has a conserved cysteine residues that ligate the ion center at the axial position. And the name P450s itself arises from the fact that if you make the ion 2 carbon monoxide complex and take a UV vis spectrum of it, there is a characteristic peak at 450 nanometer. And I think the general structures of the family is pretty well conserved. It contains a lot of, it has very high alpha helix content, as you can see from the crystal structure here. And one important feature of the P450s is that they require a suitable reductase partner to function. And different P450s have different domain organizations, and they can be classified to different classes depending on the types of domain organization. So we'll start with this class 1 system that has 3 protein components. It consists of the heme domain, which contains the cofactor, and then 2 different proteins to help with the electron transfer. So you have the iron sulfur protein and a FAD domain, and FAD stands for flavin adenine dinucleotide. We will see the structure of this cofactor in the next slide. And in this system, the electron transfer will begin by the use of NADH or NADPH by the FAD domain, and this would trigger 1 electron reduction, and this 1 electron would then be transferred sequentially to the iron sulfur domain, which then transfer the electron to the heme domain. In contrast to the class 1 system, there is the class 2 system, which does not contain the iron sulfur protein, and instead it has 2 different flavin-dependent domains consisting of the FMN and FAD domains. So both have flavins, but it has different stuff attached to the flavin, as you can see, as you will see in the next slide. And I should also mention that there are also new electron transfer mechanisms that have been recently discovered in the P450s. One of the key questions that people were really intrigued to address about these enzymes is the mechanism by which it can regulate the generation of the active iron oxo species. This is a very high energy species, and if you don't regulate the generation of the species, you will risk inactivating the enzyme by just simply oxidizing some of the residues in the active site. And nature actually has evolved a very sophisticated mechanism to help time the generation of the species. And I guess this is commonly referred to gating mechanism. So the catalytic cycle begins with this iron 3 species, and in its resting state, it has water molecule bound to it. And the first thing that happens is substrate binding to displace the water molecule. And this substrate binding actually induces spin shift in the ion tree center. And it changes from low spin to high spin. And this change in spin state actually increases the redox potential of the ion center by about 100 millivolts, and makes it more electropositive and makes it easier to reduce. And now this the redox potential of the high spin ion is more compatible with the reduction potential of flavins. Okay, so now that you've reduced the ion 3 to, you have changed the spin state, it allows the ion 3 species to be reduced to the ion 2 species. This is also known as the ferrous species. So you can see here that the electrons are delivered sequentially one by one, and it's also coupled together with oxygen activation. So first you reduce the ion 3 species by 1 electron. This is then followed by oxygen binding to generate the following ferric superoxo species. This is then followed by a proton-coupled electron transfer to generate the following ferric hydroperoxo species. This is also known as compound 0 in the literature. You can also generate this species by this peroxide shunt pathway. If you react the ion 3 species with hydrogen peroxide, you will generate this species, but this is more aggressive in nature. Starting from this hydroperoxo species, you can this can undergo protonation that triggers loss of water to generate compound 1, which is the active species in the oxidation. And the nature of compound 1 was a subject of intense study in the literature. There's a whole slew of papers that try to characterize compound 1. And I think the definitive study is done here in Science in 2010 by Mike Green. I guess if you also want to feel insignificant about your achievement as an undergrad, you can also refer to this paper, because the first author was actually an undergrad in Mike Green's lab, and he was the only author on the study. So imagine that undergrad with a first author science paper. Anyway, going back to the topic, starting from compound 1, the iron oxo species, I guess I should cover the nature of compound 1 itself. So it's an iron 4 oxo radical cation, and the radical cation nature is still localized to the porphyrin ring system. And this 2 studies here show that the cysteine ligation is important in the reactivity of compound 1. Specifically, cysteine ligation would increase the basicity for hydrogen atom abstraction. And I guess one way to think of it is that you can think of hydrogen atom abstraction as consisting of proton and electron transfer. So if you increase the basicity of the iron oxo species, then it would facilitate proton transfer to the species here. Okay, so starting from hydrogen atom abstraction, you will generate the following substrate radical species that can then undergo a radical rebound with compound 2, and this would generate the hydroxylated product and regenerate the ion 3 species that can then undergo another round of reaction. So that is the mechanism of the oxidation itself. So let's take a brief look at the electron transfer process. I mentioned before that there are 2 different types of flavin species in the reductase domain. So this would be the FMN and the FAD domain. FMN stands for flavin nucleotide, flavin mononucleotide, and this is the structure of FMN, and FAD stands for flavin adenine dinucleotide, and this is the structure of FAD. I make a mistake here. There should be this should be a diphosphate linkage. That's why it says it's dinucleotide, and you can see that there's a adenine nucleobase here, and that's how the name comes about. In terms of the reduction process, I mentioned before that this undergoes sequential one electron transfer. So you start from the oxidized flavin species, and this can undergo one electron reduction to generate the following FMN semiquinone species. And in theory, this can undergo another reduction to the hydroquinone species, but it's always thought that the hydroquinone species is not capable to reduce iron center. So I believe that in the entire electron transfer chain, you never generate the hydroquinone species. One of the most well-studied P450s is the P450 BM3. This is the most well-studied because the reductase domain and the heme domain is fused together. So it makes it a lot easier to study. And so this enzyme has been the subject of many mechanistic studies and also biotechnological applications. This paper here describes the study on the electron transfer cycle in P450 BM3. And the process goes like this. You start with, I guess let's start with the binding of NADPH to the reductase domain. Upon binding, you will then transfer hydride from NADPH to the FAD domain. And at this point, you displace NADP plus with NADPH so that you have a new molecule of NADPH in the reductase domain. This also triggers a reduction, a one electron reduction of the FMN domain to the semiquinone species and generate the following FAD radical. And then at this point, FMN semiquinone can transfer the electron to iron 3 to generate an iron 2 species. This is then followed by another single electron transfer from the FAD domain to FMN domain to regenerate the FMN semiquinone species. And at this point, we'll get back to the oxygen binding event that I described before. And then I guess after oxygen binding, you can eventually perform another single electron transfer from the FMN semiquinone to generate the iron oxo species. This is then followed by oxidation and then regenerate the catalytic cycle. So you can see here that there is one important feature in the enzyme, and that there's a sequential electron transfer that's carefully choreographed so that it can couple with oxygen activation for productive catalysis. And this is important so that you don't risk any inactivate or minimize inactivation of the enzyme. Another thing to consider is the protein dynamics in the electron transfer process. This is a schematic that shows how it happens in a macromolecular level. It's always thought that the FMN domain actually would undergo a large-scale movement through the movement of the hinged region here, so that it would swing from the FAD domain to the heme domain to make the electron transfer process more favorable. So I hope you'll see from this example that enzymes are very sophisticated catalysts, and there's a lot of protein dynamics that are involved in the process to enable productive catalysis. So let's move on to the types of reactions that can be catalyzed by the P450s. Of course, they can perform various types of oxygen transfers, and this range from hydroxylation reaction to desaturation, and you can also generate nitro product by oxidation of amines. Some P450s can also catalyze epoxidation of double bonds. And the types of hydroxylation is not limited to just sp3ch hydroxylation. You can also perform aromatic hydroxylation to convert benzenes to phenols. And there is also there are also several P450s that can catalyze biaryl couplings between two electron-rich aromatics. And the key species for the oxidation itself is not limited to compound 1. Several other intermediates in the catalytic cycle can also be important for catalysis. So for example, compound 0 can perform epoxidation through the arrow pushing mechanism shown here. And then finally, in this rather interesting nitration reaction, it's been proposed that the ferric superoxide species plays an important role in generating the following peroxynitride intermediate that can then decompose to compound 1 and generate the following nitrating species that can then nitrate the substrate here. And you can learn more about this in this Nature Chem Bio paper. In terms of the hydroxylation reaction itself, I think the particular enzyme that has been studied the most for applications in both synthetic chemistry and biotechnology is the P450 BM3. And I mentioned before that it has that its reductase domain is fused with its heme domain, and this makes it a lot more convenient to study and also use in various applications. Its native function is to hydroxylate long-chain fatty acid, but subsequent engineering work, mainly in the labs of Francis Arnold and Manfred Reeds, have successfully converted the function of this enzyme from long-chain fatty acid hydroxylase to become a hydroxylase that can act on a wide range of organic substrates. And this slide provides a brief summary of the types of substrates that have been reported for the hydroxylation reaction. And I've specifically chosen molecules that you might be interested in because of their potential utility in natural product synthesis, I guess. And virtually almost every residue in the enzyme has been targeted for mutagenesis. And you can consult this very detailed review here by Luet Wong in ChemSocRef if you wanna see the summary of the types of effects that would happen for mutations at certain residues. We're gonna move on to the non-heme system, and I'm gonna start with the iron and alpha-ketoglutarate dependent dioxygenases. And this enzyme is characterized by the presence of 2 histidine and 1 carboxylate ion binding motif. And as the name suggests, it also contains a ketoglutarate acid co-substrate in the active site that binds through this binding mode. In terms of the reaction cycle, the mechanism is shown here. Substrate binding will displace water from the active site, and this would then trigger oxygen activation. And this is a pretty interesting process because nature actually couples the oxygen activation process with a decarboxylation event that converts ketoglutarate acid to succinate. And this is proposed to occur through attack of the hydroperoxide species to the carbonyl group here. This can then decompose to generate the iron 4 oxo species. From here, the reaction proceeds through the same mechanism as P450 hydroxylation. So it would go through hydrogen atom abstraction from the substrate that's followed by radical rebound to generate the hydroxylation product. This enzyme superfamily actually has been known for a long time. The first discovery was in the hydroxylation of proline residues in collagen. And if you're familiar with the work of Ron Rains, you might know that this hydroxylation is important in maintaining the, I guess, the secondary and tertiary structure of collagens. This is a very diverse enzyme superfamily, and there's a whole slew of hydroxylation reactions that can be catalyzed by different members of the family, ranging from amino acid hydroxylations to hydroxylations of indole alkaloids. This is the product here is an intermediate in the biosynthesis of phenblastin. And then you can also hydroxylate terpenes. And this is a recent example from 2019 that was done at Scripps, Florida by our colleague here, Professor Ben Shen. This reaction here is involved in the biosynthesis of potentamycin. In addition to hydroxylation, the alpha-Kgs can also catalyze halogenation reactions. And in order to perform halogenation, the carboxylate ligand that's binding to the iron is actually mutated to alanine or glycine residue, and this would free up one coordination site on the iron, and this empty coordination site would allow binding of a halide anion to the iron center. So the catalytic cycle proceeds through the same reaction, and eventually you will arrive at this iron oxide species. This can then perform the same hydrogen atom abstraction. And now the substrate radical in the halogenation reaction can undergo a selective radical rebound with the chloride ligand instead of the hydroxide. And this leads to the generation of the chlorinated product. And it's been shown that substrate positioning is important to govern the selectivity of the halogenation reaction. And this was done through some mechanistic studies on the first halogenase to be, alpha-Kg halogenase to be discovered, which is SIRB2. This enzyme acts on threonine that's been, ligated to a carrier protein called SIRB1, and I believe this is involved in the biosynthesis of syringomycin, which is a non-ribosomal peptide natural product. So in this reaction, SIRB2 would catalyze the chlorination of this metal group on SIRB1 bound threonine to provide the following product. One important thing to note is that if you switch the substrate from threonine to norvaline, you actually would get primarily just the hydroxylation. And this, shows that the identity of the substrate is important so that the enzyme is able to position the substrate in the right orientation for halogenation reaction. Otherwise, you will end up with other types of reaction. And the Bollinger and Krebs group at Penn State has also done very detailed mechanistic studies with EPR to explain the geometry of the reaction that will allow selective chlorination. And this also has been, further supported with some QMMM studies that's reported in this ACS catalysis paper. In addition to halogenation, the Bollinger-Krebs group has also shown that you can use these halogenases to catalyze azidation or nitration reaction. So essentially, the reaction would proceed through the same reaction mechanism, but now instead of having a chloride ligand, you will have either an azide or a nitrate ligand, and the substrate radical can then undergo rebound with that ligand to generate either the acidated or the nitrated product. This is the first example of this type of acidation or nitration reaction, but subsequent work from several other labs like Michelle Chang have shown that you can also use other types of alpha-Kg halogenases to do the same acidation or nitration reaction. In addition to this halogenase that acts on amino acid, there are several halogenases that have also been characterized that act on different types of substrate. This is one example that's reported from the wilbitinolinone biosynthesis. An enzyme called WEL-O5 can catalyze chlorination in the cyclohexane motif of this precursor to wilbitinolinone. And there's also a homolog of this enzyme called AM-O5 that shares pretty high sequence identity with WEL-O5. And I believe this Angwanta paper showed that this enzyme AM-O5 has less stringent substrate specificity compared to WEL-O5. And later on, they showed that if you perform, if you generate a chimera between WEL-O5 and AM-O5, you can alter the reduced selectivity of the halogenation reaction. Now, if you look at the active site geometry in the halogenases and compare that to the hydroxylases, you might have the naive idea that you can simply convert hydroxylase to halogenase by simply performing mutation at the glutamate or aspartate residue, and then add a lot of chloride in the reaction to trigger halogenation. But in reality, this conversion is not trivial. And this is illustrated by this work reported in 2009 by Ron Rains. And here they tried to mutate the, I guess this is the, they mutate aspartate 414 on proline for hydroxylase to alanine or glycine in the hope that they would be able to catalyze chlorination with the resulting mutant. But instead, when they performed this swapping, they got an enzyme that's entirely non-functional. So it does not perform hydroxylation and it does not perform chlorination either. I think the first success in converting halogenase to hydroxylase or vice versa was reported in this Nature Chem Bio paper in 2016. I believe this was done by Amy Boll at Penn State. And this was done by performing rational mutagenesis based on soft crystal structure of the enzyme. By analyzing the crystal structure, they were able to predict the residues that might be important in governing the selectivity of the reaction, and they were able to perform single mutation that I guess confers hydroxylation activity on WEL-O5. They still produce a mixture of hydroxylation and chlorination, but I think this is one of the first report of successful conversion of halogenase to hydroxylase on the native structure, I guess. Okay, the last superfamily of hydroxylases that I'll be covering today is this risky dioxygenases. This superfamily is characterized by the presence of this active site. So it has this non-heme center that contains a 2-histidine and 1 carboxylate iron binding motif. So rather similar to the alpha-Kgs, but in this case, the iron center does not bind to ketoglutarate acid. And in addition to this iron center, there's also this risky center that has this 2 iron 2 sulfur cluster that's implicated in the electron transfer. This is first identified in the degradation of aromatic compounds by a Pseudomonas species, and this degradation takes place through the hydroxylation of the arene ring. Later on, this enzyme is further identified to be a 3-component system, naphthalene or toluene dioxygenase. And the component is shown here. So it has a flavin-dependent reductase, ferredoxin reductase, and this ferredoxin has 2 iron 2 sulfur center. And finally, you have the oxygenase domain that has the iron that has this risky center. In terms of the function of the dioxygenase itself, as the name suggests, it performs the hydroxylation of naphthalenes and toluenes, and as well as several other aromatics. I guess I should also mention that both naphthalene and toluene dioxygenase has been utilized several times in biocatalysis through some of the work by Thomas Hadlicki. And he has also shown the applications of this reaction in the synthesis of some complex natural products. And I believe Andy Myers also used the same arene dihydroxylation in his synthesis of tetracycline. So let's move on to the electron transfer process. As I mentioned, risky dioxygenases contains 3 components, and the electron transfer happens as follows. So the flavin-dependent reductase would consume NADH or NADPH to generate the reduced species, and this can then transfer electron to the ferredoxin component that in turn performs subsequent electron transfer to the ion center. So I guess it's rather similar to the P450s, where you have sequential electron transfer from different domains to facilitate the oxidation reaction. The reaction mechanism itself is not as well understood, and some of the challenges here are as follow. This is a multi-component system, so it's not always easy to reconstitute. And as I mentioned before, iron sulfur clusters are typically oxygen sensitive, so it can be rather difficult to work with these enzymes in purified form. And finally, there's a lack of chromophore for spectroscopic studies, so it's not as easy to interpret some of the results of spectroscopic studies. Nevertheless, the reaction mechanism has been postulated, and this goes as follows. So oxygen activation would generate the following bidentate hydroperoxide species. And from here, there are 2 possible reaction mechanisms that can take place. You can either perform the reaction in a more sequential fashion, where you decompose the bidentate hydroperoxide species to the following iron oxo, which can then trigger like a sequential hydroxylation of the double bond to generate the product. Or you can more directly couple the decomposition of the hydroperoxide with CO bond formation. In both cases, you'll finally generate the following hydroxylate product that would then be released and then regenerate the resting state of the enzyme. Similar to the P450s, the reactions that can be catalyzed by the risky enzymes are not just limited to hydroxylation of CH bonds. There's an enzyme called PRND, which belongs to the risky superfamily that's been shown to catalyze conversion of amines to nitro group in pyrrolonitrine biosynthesis. This is work that was done by Weimin Zhao at UIUC, and his lab has also done several subsequent studies on this enzyme. They've reported that it has some degree of substrate promiscuity, and it can catalyze the oxidation of several other types of anilines. And they have also performed mechanistic studies that indicate the presence of various partially oxidized intermediates en route to the nitro product formation. And then finally, they've also been able to perform a little bit of engineering on this enzyme to improve the catalytic efficiency in the nitration reaction itself. You can see that they were able to improve the kinetic parameters for the oxidation reaction by almost tenfold here. And then finally, there are also several risky enzymes that can catalyze CC coupling reactions. And this can be found in the biosynthesis of the protegiosine family of natural products. This family might have been covered by Phil in the heterocycles class. In any case, they all share the same tripyrrole motif with this long chain alkane. And in the biosynthesis, nature actually takes one common precursor called undecyl protegiosine, and different enzymes can catalyze CC bond formation to form different ring sizes to generate different types of product, namely butyl cycloheptylprotegiosine, metacycloprotegiosine, marineosine, and streptorubin. And it's been proposed that, these are the names of the enzymes that catalyze the actual transformation to the respective products. So you have REDG, MCPG, and MARG. And it's been proposed that the reaction would proceed through the formation of radical on the long chain alkane motif that can then undergo CC bond formation with the parole ring to generate the product. And I guess in this case, the difference in the active site of the respective enzymes would allow selective radical generation at different position on the alkyl chain, leading to specific formation of different types of ring sizes in the product. Any questions about hydroxylation at this point before we move on to radical SAM enzyme? So let's move on to radical SAMs. As the name suggests, this enzyme superfamily uses SAM, or S-adenosylmethionine, in its reaction. And the other important component in the reaction is 4-iron-4 sulfur cluster that's present in all members of the family. And these two components can combine together in the active site of this enzyme to form the following complex. And I believe that this species has been characterized in one of the enzymes that have been studied before. There's an extensive review on this superfamily in this ChemRef paper. If you want to read up on the all the types of reactions that can be catalyzed by this enzyme superfamily. But in general, the reaction mechanism is the same for many members of the family. So you start from this SAM iron sulfur 4-iron-4 sulfur complex that can then undergo one electron reduction. And then this is then followed by I guess, homolysis of one of the CS bond in SAM to generate deoxyadenylyl radical and methionine that's still bound to the iron sulfur center. And this is usually the active species that can perform other types of reactions. I should also note that the same deoxyadenylyl radical intermediate can also be generated from the decomposition of adenylcobalamin, whose structure is shown here. And there are several radical SAM enzymes that can work in cooperation with cobalamin to perform radical metallation reaction. So in this case, you'll have your deoxyadenylyl radical react with your substrate to generate a substrate radical. And in the other cycle of the reaction, you have cobalamin that can react with SAM to generate methylcobalamin, and this would become the active metallation species that would result in a net CH methylation of in the reaction. So let's talk about the enzyme in a little bit more details. There's several energetic considerations for the radical and for the reduction of radical SAM enzymes. So it's been calculated that the reduction potential of free SAM itself is about minus 1800 millivolts. So it's very hard to reduce. And at the same time, the reduction potential of 4-iron-4 sulfur cluster itself is only about minus 500 to minus 600 millivolts. So if you consider these two numbers, radical generation from the combination of these two species is energetically unfavored when considered in isolation. And because of this consideration, people have thought that the binding mode of the complex in the active site plays some role in altering the redox couple to make the radical generation event more favorable. The other thing that we should also consider is in the homolysis of the SAM species itself. If you look at the structure of SAM, there are 3 different CH bonds that can undergo homolysis. So one question that people were interested in addressing is the role of the active site in governing which CH bond would be broken in the reaction. And spectroscopic studies, including crystal structure studies, have suggested that in this binding mode, there's a direct orbital overlap between the iron sulfur bond and the sulfonium atom here. And you can see that in this case, there's the orbital overlap between the the sigma star of the CS bond that's connected to the adenosyl motif with the iron sulfur bond. And people believe that this overlap is important in facilitating selective cleavage to generate the following deoxyadenosyl radical. So I guess there could be some type of direct electron transfer between the iron sulfur center and the anti-bonding orbital of this carbon sulfur bond that's connected to the adenosyl motif here. Now let's take a look at some reactions that can be catalyzed by radical SAMs. Here we're gonna first take a look at sulfur insertion reaction. This is involved in the biosynthesis of biotin. Biotin itself is a component of vitamin B7, which is involved in a range of metabolic processes. And this biosynthesis is catalyzed by an enzyme called BioB. The constitution of BioB showed that it has 1 4-iron-4 sulfur and 1 2-iron-2 sulfur cluster per enzyme monomer. And the 4-iron-4 sulfur cluster is retained throughout the reaction, but the 2-iron-2 sulfur component is degraded in the reaction. And based on this information, it's likely that the 2-iron-2 sulfur would act as the source of sulfur in biotin. And it's believed that the reaction proceeds through the following mechanism. So you would first generate the adenosyl radical from SAM through the reaction mechanism that I've shown in the previous slide. And this radical species can then perform CH abstraction on this metal group to generate the following radical intermediate that can then react with the 2-iron-2 sulfur cluster to generate the following CS bond. Another homolysis event would then generate the same adenosyl radical to generate another radical species on the other side of the substrate that can then perform the same CS bond formation to generate this 5-membered ring. And in the process, it also leads to degradation of the 2-iron-2 sulfur cluster with the following product. Similar reactivity can also be observed in the biosynthesis of lipoic acid. And this is the structure of lipoic acid here. And this reaction is catalyzed by an enzyme called LipA. And the reaction mechanism is similar to BioB. So you would generate a substrate radical that can then undergo CS bond formation. But in this case, the enzyme uses a 4-iron-4 sulfur cluster for the CS bond formation. In addition,
could you comment on the selectivity for the adenosyl radical going after the, I guess, what looks like the strongest CH bond in the substrate? Is that just how it's oriented in the
I
binding
think in a lot of these enzymatic reactions, the active site of the enzyme is able to orient the substrate in such a way that only one particular position is exposed for reaction. In addition to CS bond formation, this superfamily can also catalyze CC coupling, and this can be found in the biosynthesis of tunicamycin. The enzyme involved is called TUNB, and the reaction proceeds through the same deoxyadenyl radical formation. This can then perform hydrogen atom abstraction on this methylene group here, and the resulting radical species can then undergo a radical addition to this sugar motif that would then, I guess, eventually to form the following intermediate that would then go on to become tunicamycin. Okay, I have about 30 minutes left, and I'm going to switch gears to artificial metal enzyme. And I guess artificial metal enzyme is broadly defined as unnatural enzymes that are derived from insertion of a catalytically competent metal cofactor into a protein scaffold. And there are several strategies that have currently been used for incorporation of metal cofactors into protein scaffold, and these are illustrated in these 4 diagrams here. So first, you can perform covalent bond formation to attach the cofactor into the active site. As this is typically done by using a nucleophilic residue in the active site to attack an electrophilic group that's been incorporated into your cofactor. You can also perform supramolecular anchoring, and this is done by exploiting the high affinity of certain scaffolds for particular substrates, and we'll see a little bit of that in a little bit. You can also perform dative bonding with the metal center itself, and this is usually done through the use of coordination of hydrogen-containing side chains such as histidines. And then finally, you can perform a simple metal substitution on existing natural metal enzymes. And this is still a highly active area of research, and there have been a lot of reviews that try to cover this topic, and you can consult these reviews listed here if you want more details on this field. The first demonstration of artificial metal enzyme catalysis was reported by George Whitesides in, I guess, almost more than 40 years ago. And this is done through the use of apidin-biotin couple. And before going to the reaction itself, I'm going to give a brief primer on biotin and apidin, or streptafidin. So this is one of the most well-known ligand-protein pair in nature. Apidin itself is the tetrameric protein, and it is capable of binding biotin with very high affinity. So the KD is, I guess, it's in the picomolar range. And so you can think of this couple as basically a covalent bond formation between the two. And the Whitesides group was able to use this feature to anchor a rhodium complex into biotin. So they were able to generate the following biotin rhodium complex that's then reacted with apidin. And they were able to use this in hydrogenation reaction, and they reported about 500 turnover number with 41% EE. But I think one of the issues with the system is that it's not readily reproducible. I think that's what Kim Jenda once told me. And I think that was one of the reasons why this technology lay dormant for a while. I guess before moving on, I also want to cover the structure of apidin and biotin. So apidin itself is a tetrameric protein, and it can bind up to 4 molecules of biotin. And the binding site for biotin itself actually lies in the protein-protein interface between the monomers of the protein. And so this makes for a pretty well-defined active site and makes it easy to engineer and mutate for certain reactions. In the late 90s, the system was revisited by several groups, and it was later shown that by using a different type of biotin rhodium complex, you can achieve very low turnover number with some degree of EE. And around the same time, Manfred Reeds also tried to develop a different system involving papain as the protein backbone that's then connected with the following rhodium complex. And they tested the system for some hydrogenation reaction, but they only observed an EE, very low EE in the reaction. So I guess they kind of conclude that it's, this is not very promising for catalysis. Following the studies though, Tom Ward at University of Basel decided to perform more detailed and systematic studies on the system in order to be able to improve it further for biocatalytic reactions. So they decided to screen a range of ligands that can bind to rhodium. And at the same time, they also perform mutagenesis on streptafidin to identify mutations that can be beneficial for catalysis, specifically for hydrogenation reaction. So through this library against library screening, they were able to identify several combinations that proved to be useful for catalysis. So they were able to obtain quantitative conversion with more than 90% EE for either the R or the S enantiomer, depending on the ligands that you use to bind rhodium. Because one drawback of the system is that the protein loading is usually still rather high, especially if you consider the typical loading range for biocatalysis. Okay, following the invention of the system, they were able to adapt the platform for other types of transition metal catalysis. This range from cross-coupling, and this was done by the use of this double mutant in the presence of the following palladium catalysts, and they were able to get 90% EE for enantioselective biaryl bond formation in this cross-coupling with 50% or 50 turnover number. Collaborative work between the Ward group and the Rovers group resulted in the adaptation of the system to CS activation reaction. This is work done by Todd Heister when he was a visiting student with Tom Ward, actually. So using the same double mutant, they were able to perform a CS activation reaction to generate the following lactam product. And this is done through the use of this biotin-linked rhodium complex. More recently, the Ward group has also been able to adapt the process to ring closing metathesis, and they also were able to demonstrate a proof of concept idea that will allow them to couple the system with in vivo directed evolution to readily improve the activity of the enzyme. And the idea is shown here. So they would have the following Krebs catalyst that will be linked to biotin, and then they will have also E. coli that will be expressing various streptafidin mutant. And they are going to use the following substrate. And the idea here is that if RCM happens, you'll generate the following coumarin product, which will be fluorescent. And they have to tweak the system further. In this case, they actually have to fuse streptafidin to a protein called OMA. OMA itself functions to direct the protein towards the periplasm where there's lower concentration of glutathione. I guess glutathione might poison Krebs catalyst, so they want to be able to export the protein scaffold to the periplasm. And the workflow is shown here. They're going to generate mutant library that would then be expressed in 96-well plate. And then after expression of the mutants, they would then spin down the cells to discard the supernatant, and then they're going to use the reaction in, I guess in this, this would be done in lysate. So they resuspend the pellet in the reaction buffer, and this is also where they would incorporate the biotin link Scripps catalyst. And then at this point, the ruthenium catalyst would be incorporated into streptavidin scaffold, but there might be some excess ruthenium complex. So it's important to do the subsequent treatment here where they discard supernatant so that they can minimize background reaction. After that, this is then ready for reaction. They can then add the substrate and then just look for wells that display fluorescent and then characterize the mutants that can result in the formation of the fluorescent product. And by doing so, they were able to identify mutants of streptavidin that can catalyze ring closing metastasis on the system. You might think that this, since to be overly engineered, it might be valid, but I guess at least it's a proof of concept that you can use the system for ring closing metastasis. So I guess in this area, I think the concept is almost always the same. You just link your metal complex of interest to biotin and then incorporate it into streptavidin and then test for reactions. And if you need to improve the reaction, you perform mutations on the so-called active site, which is formed in the interface between the monomers of streptavidin in the tetramer. In addition to the streptavidin system, several groups have reported other types of scaffold for the construction of artificial metal enzyme. This is one example from the Lewis group, now at Indiana University. They chose the following scaffold called prolyl oligopeptidase to construct their artificial metal enzyme. This scaffold is chosen because of its cylindrical shape and also its large internal volume. And they hypothesized that these features would allow suitable anchoring of different cofactors, and they decide to anchor a dyrodium complex with the goal of performing different types of carbene and nitrene chemistry. And their idea here is to link the rhodium complex with this cyclooctane so that, and this can then be anchored into the active site through the use of a click chemistry. And to do so, they have to incorporate acetophenylalanine residue into the active site, and this is done through the use of amber suppression that's been developed by Pete Schultz. And over the years, I guess they've done different iterations of the reaction, and I think the most recent report is in the use of the system for cyclopropionation reaction. And I guess and also CS insertion reaction. And before going to the result, maybe I should briefly describe the workflow that they developed to optimize the reaction. So they would perform error-prone mutagenesis on the scaffold itself. This is done through error-prone PCR, and then they would then incorporate the fragment into the gene through the use of Gibson assembly. And then now that you have your variant library, you can co-transform E. coli with this variants and also with the plasmid that's required for the incorporation of acetophenylalanine. Okay, so now you express everything in 96 well plate per normal, and then at this point, after you prepare the lysate, you can introduce the cofactor by simply adding a solution of the rhodium complex. But at this point, the click reaction should happen to generate the covalent bond between the rhodium complex and the active site of the protein. But there might be some free rhodium that's present in the system, so they have to get rid of it, and this is done through the use of solid phase extraction, so that you can pull any excess rhodium species from the lysate. Okay, so now that you've done this step, hopefully you will get rid of all the excess rhodium that might interfere with the reaction, and now you can screen for reaction. And this is done through, rather brute force HPLC or SFC screening, and then you identify hits and then repeat the same process like a typical directed evolution experiment. And by using this process, they were able to identify several variants that can catalyze selective cyclopropionation of styrenes, and also CS insertion on this cyclohexadiene compound here. The Ruffles group has also used a different type of scaffold for artificial metal enzyme creation. And in this case, they use a protein called LMRR, which stands for Lactococcal multi-drug resistant regulator. And some of the features of this protein are similar to Jarrett's system and also the streptavidin system. It's a homodimeric protein, and it also has a large hydrophobic core. So they hypothesized that this would be able to accommodate hydrophobic substrates for reactions. In their design, they decided to incorporate the following by pyridine ligand. This is done through the use of amber suppression. And they showed that by using the system and adding copper salt to generate the copper complex, they were able to perform enantioselective 1,4 addition with indoles like this. Later on, they also developed an alternative strategy that does not require the use of amber suppression. In this case, they rely on hydrophobic interaction between this fen ligand and the 2 tryptophan residues in the active site, so that they can bind this fen ligand, and then you can use it for the same type of reaction in the presence of copper salt. And finally, there are several other types of anchoring strategies to incorporate different metal complexes to enzymes. And this is done through, anchoring through attack of nucleophilic residues in the protein backbone by linking the complex with an electrophilic motif. I guess this is kind of like using Bancroft's ABPP platform, but for catalysis. So for example, you can use this phosphonate electrophile to incorporate the following rhodium NHC carbene complex into cutanase. And then you can also use alpha haloketones to incorporate this ruthenium complex into chymotrypsin. Okay. The last strategy that I'm going to cover for artificial metal enzyme creation is the use of metal substitution. And for this, I'm going to cover some work by John Hartwig for the use of metal substitution in myoglobin to catalyze carbene transfer reaction. And the idea for metal substitution is shown here. They're going to use myoglobin as the scaffold, and myoglobin itself is a heme-containing protein. And so the key here is to be able to express myoglobin such that it won't contain any iron. And this is done by the use of low iron expression conditions. So you can grow the cells in minimal media that does not contain iron. And by doing that, you will express apomyoglobin that will not contain heme. So at this point, you can then purify the resulting protein product. So you'll get the apoprotein, and then you can incorporate different types of metal porphyrins into the system. So they tested a wide range of metal substitutions. And you can see that, you can see the colors here. And then they first tested the reactions of the different metal substituted myoglobins for carbon transfer reaction with the substrate. And they actually observed that iridium porphyrin is the best system for this reaction with pretty high turnover number. And following this discovery, they have been using this iridium porphyrin system for subsequent types of carbon reactions, carbon transfer reactions. They've also done some mutagenesis to tune the selectivity of the reaction. This is just the directed evolution workflow that they use for optimization of the reaction. I guess I should note that in all of, I believe that these are all done through site-directed mutagenesis, meaning that you make specific mutation and not generate a library. And I believe that for each one of these variants, they actually would make the mutant and then purify the enzyme and then test that for reaction. So it's quite a lot of work. But in any case, they were able to identify several variants that actually gave divergent selectivity profile, where they were able to generate different enantiomers of the products. And these are just some examples of the products that they can form in the reaction. And I believe at that time, this was the first report of being able to perform cyclopropionation on unactivated alkenes with artificial metal enzyme, as well as CH insertion reaction. Later on, they switched to a different protein scaffold. In this case, they use a P450 called CYP119 and then incorporate iridium porphyrin into this P450. And they were able to observe better kinetics and reaction scope by using this system. All right, for the final part of my talk, I'm gonna talk about some of the work that's been done by the Arnold group in repurposing heme proteins for carbon-nitrogen transfer reaction. And this is done without using metal substitution. So in all of these cases, they use the native heme cofactor in the reaction. The reaction cycle, in a broad sense, is shown here. You start from the, I guess it's kind of like hijacking the catalytic cycle of P450, but instead of forming an iron oxo species, you're going to generate either an iron carbenoid or iron nitrenoid by reacting the iron 2 species with either diacyl compound or an A site. There's some precedents for this type of reactivity in the organometallic literature. In 1995, Tom Kodadek, then at UT Austin, reported the use of the following iron porphyrin for cyclopropionation of styrenes with very high turnover numbers. And Ron Breslow also showed in 1985 that you can use rabbit liver P450 to catalyze CS amination from this iminoiodinane species, albeit with very low turnover number. So based on this precedence, they believe that they can generate this, the same type of cyclopropionation or amination reaction. The first report was done in 2013, where they showed that they, by screening different P450 variants, they were able to perform cyclopropionation of olefins in an enantioselective fashion. And they also showed that you can readily modulate the EE and the cis trans selectivity profile in the reaction by performing mutagenesis. There's also one key discovery that was made here, which is the substitution of the cysteine axial ligand to serine, and this allows the reaction to be performed in whole cell. They found that by doing this substitution, they increased the redox potential of the iron 3 to iron 2 to be more electropositive, so making it easier to reduce in under physiological conditions, and that's why they were able to use this serine ligated enzyme for cyclopropionation in whole cells. And then subsequently, they also showed that the reaction is not just limited to cyclopropionation. You can perform NH insertion to anilines. This reaction proceeds with a pretty good turnover number, although it was completely racemic. I think only recently they were able to show that the reaction can be done in an enantioselective fashion. In addition to carbon transfer reaction, they also showed that you can perform nitrogen transfer reaction. First, they started with this intramolecular system that's very similar to Breslow's system. Using the same P411 BM3 variant, they can get about 400 turnover number for amination. They also observe, I think, pretty good enantioselectivity for this reaction here. They subsequently showed that the nitrogen transfer is not just limited to intramolecular cases, and by using sulfamidation as a case study, they were able to perform intermolecular amination reaction. Since this discovery, they have been able to expand the type of reactions that can be performed with this platform. And I think more recently, I think you're starting to see that the types of reactions that can be performed are becoming more and more difficult. So now they can perform enantioselective CS insertion or CS amination. So this is a recent report from 2 years ago in the use of pre-engineered P411 variant for CS insertion into activated CS bonds. You can, I think here, I think there is more than 15 rounds of evolution needed to convert to really optimize the activity of this variant. But the resulting enzyme is highly active for CS insertion on several different types of substrate, ranging from benzylic substrates to allylic substrates, and also insertion into CS bonds that are adjacent to heteroatoms like amines like this. You can also perform enantioselective intermolecular CS amination. So the first demonstration was done through the use of tosyl azide as the nitrogen source. And for this report here, I believe that these are all limited to benzylic CH bonds, but nevertheless, they were able to get pretty high turnover number with reasonably high EE for the amination product. And more recently, they've been exploring the use of alternative nitrogen source. They were able to use the following p-folate salt. I believe this was popularized by the work of Bill Morandi, but the Arnold group showed that you can also use this reagent for amination reaction with their P411 variants. And I guess the nice thing about the use of this reagent is that you would end up with free amines in your product instead of the tosyl amine. And they showed that using the system, they can perform enantioselective benzylic or allylic CS amination on a range of substrates. And these are just some of the products that they've reported using this system. Okay, I think that's the end of my lecture. Any questions about any of the system? I'll be happy to answer them.